On the behalf of the complete organizing committee, I warmly welcome you all to session 25, Environment Track 4 of International Conference of Energy, Water and Environment ICEWE 2021.
Our session chair is Dr. Tayyaba Asif. In this session, there are a total of 5 talks. At the end of each talk, we will be having question and answer session. The audience is requested to write the questions in the chat box during the talk. At the end of each presentation, the session manager will ask the requested question from the presenter. Without any further delay, let me introduce our session chair, Dr. Tayyaba Asif. She is working as a scientific officer in Aquatic Diagnostic and Research Center, Bahria University, Karachi campus. The area of her research is crustacean waste, wastewater extraction, and application of biopolymers from crustacean origins. So it's an honor to have you with us, Dr. Tayyaba Asif. I humbly request you to please share your thoughts.
Assalamu alaikum. I'm Dr. Tayyaba Asif, working as a scientific officer at Aquatic Diagnostic and Research Center, Bahria University, Karachi campus. Today I'm giving a presentation for ICEWE, and the topic of my presentation is treatment of fishery waste through chitinase produced by Glutamicibacter urotoxodons Kibji IB 41. In this presentation, I will focus on the marine-based waste. This marine-based waste was generated after the processing of crustacean waste, and it increases the marine pollution because this waste rich in protein, calcium carbonate, and chitin. Chitin, which is a biopolymer, but it is highly insoluble in water, and due to this nature, it increases the water pollution. And due to the rich enrichment of this waste in protein nature, this waste increases the chances of any hazardous or potential pandemic, because the microorganism which utilizes these kind of waste may increase their potential to cause different type of diseases. In order to that, an enzyme which is known as chitinase used for the hydrolysis of this marine-based waste. Chitinase is basically an hydrolytic enzyme which is used to cleave the chitin into its biopolymers. The chitinase is important because it is used and known for its industrial application, and it is used for the degradation of chitin-based waste. In the current study, the overall methodology was focused on the sampling of microorganisms from different indigenous sources for the production and purification of chitinase. In the second step, indigenous microorganisms were identified using polyphysic approach, and the organisms were also screened for the production of chitinase. After that, the optimization of chitinase was performed, followed by the purification of chitinase. The purified chitinase were further characterized for finding out its ability to be utilized in the industrial applications. The time duration used for 2 to 6 hours, and the temperature was variable in the range of 15 to 55 degrees Celsius, while different buffers were used for obtaining its potential at different pH. These are the overall results, in which we obtained a novel microorganism, and it was observed that it is a gram-positive organism. This microorganism has ability to hydrolyze chitin. It was also observed the clear hydrolytic zone. This microorganism was further identified using polyphasic approach. Polyphasic approach uses microbial testing, biochemical testing, and molecular testing for the identification of microorganisms. The microorganism was further identified as novel microbacterium known as Glutamicibacter urotoxidans. After that, the quantitative screening was also performed, in which it was observed that among two different isolate B, that is Glutamicibacter urotoxidans, KIB41, was found to be the hyperproducer of chitinase. And on the basis of that, this microorganism was further used during the study. After that, optimization of different fermentation parameters were performed, and in which it was observed that, as I mentioned earlier, that chitin is highly insoluble in water, and due to this nature, it was observed that it takes a lot of time by the organism to hydrolyze this biopolymer. And after 72 hours, a peak was observed in terms of chitinase production. While this microorganism has ability to hydrolyze chitin at 25 degrees Celsius, which is a main lead for this research. It was also observed that the microorganism has ability to ferment chitin with the help of chitinase at pH 7. So the overall production parameters suggested that the microorganism produced chitinase after 72 hours of production time at 25 degrees Celsius with pH 7. This suggested that the microorganism may able to grow at normal temperature, the temperature which is usually near the water bodies. After that, the protein was purified through gradient precipitation technique, in which ammonium sulfate salt was used, and it was observed that 47.9% yield was obtained after first step or initial step of precipitation. After that, different approaches were applied for the desalting of the enzyme. And it was observed that 50% and 53.2% enzyme yield were obtained. While final purification was performed using CL6B column, and after that 63.9% enzyme purification was obtained. Followed by the characterization. Characterization basically performed for the analysis of enzyme ability to utilize it in industry. It was observed that chitinase hydrolyzed chitin after 2 hours of incubation time. While during the temperature kinetics, it was observed that a variable range of kinetic activity, chitinase activities were observed. From 15 degrees Celsius to 35 degrees Celsius, this enzyme was activated. While after that, a sharp decline was observed in its activity. This suggested that this enzyme might use for the wastewater treatment purposes. This enzyme acts at pH 7 with the help by using potassium phosphate buffer. It was observed after using potassium phosphate buffer. So the study concluded as the chitinase, which was produced by Glutamicibacter urotoxidans KIB41, which is a novel bacterium and not reported earlier for the production of chitinase. Chitinase was purified using CL6B column. Chitinase can be used for the waste management as it works around 15 degrees Celsius and more. Chitinase could also be used as a biocontrol agent, and it was also observed during the literature review that chitinase could be used for the treatment of asthma. I would like to acknowledge HEC for the grant of this project, University of Karachi as my parent university, Dr. A.Q. Khan Institute of Biotechnology and Genetic Engineering for overall funding of this research and providing lab facilities, and Bahria University for giving me opportunity to present myself in ICEW. These are the references used for the for this abstract and presentation making. Thank you.
Thank you very much, Dr. Tayyaba Asif. If the audience have any question, you can ask in the chat box.
Okay, Dr. Tayyaba Asif is not available online. So without any further delay, I am moving towards the next speaker.
Okay, thank you very much, Dr. Tayyaba, for being with us. Now, quickly moving forward, I am moving towards the next speaker. Her name is Javeria Ahad, and she is from Pias, Islamabad. Her title of presentation is Parametric Study of Nuclear Power Plant Emergency Venting System.
Assalamualaikum, this is Javeria from Pias, and today I am presenting my research work on parametric study of emergency venting system for nuclear power plants. When a nuclear accident takes place, it can be due to any reason. It can be due to a malfunctioning of an instrument or a system, or it can be due to some internal leakages, etc. Whatever the reason may be. When a nuclear accident takes place, it often results in production of steam inside this core. This steam formation leads to increase in pressure inside this core, and when this pressure exceeds beyond the design limit of the system, it results in massive explosion. Now, explosion in nuclear power plant is different from the other explosions because all the radioactivity that is cooped inside this core will end up spreading into the environment and causing havoc on people and environment for ages. As it can be seen here, this is steam formation, and all the radioactivity that is present inside the core is being released into the environment as a result of explosion. Now, this is actual footage of Fukushima accident from Japan that took place in 2011, and as a result of this accident, a massive explosion took place, spreading all the radioactivity into the environment. This is the spread of radioactive iodine into the environment. This footage is also from Fukushima accident. To avoid this catastrophe, a nuclear safety system was specifically designed to keep the nuclear power plant and people safe. This system was named Filtered Containment Venting System, and the purpose of this system was that when a nuclear accident takes place and pressure begins to build up above a set point inside the nuclear power plant, the overpressurization condition is maintained by letting out some of the containment air via this pathway into this huge tank. Now, the air coming from the nuclear power plant is full of radioactive products, so we cannot let this air out directly into the environment. So this tank contains chemical solutions and many scrubbers that can help remove radioactive products. Now, after the removal of radioactive products, the air is then vented out into the environment. Among the list of the radioactive products, the radioactive iodine is the most hazardous one because it can cause thyroid cancer among other hazards. So it is very important to do proper research on the removal efficiency of iodine to get maximum removal efficiency and also to ensure the safety of people and environment. An indigenous lab-scale setup of this system was actually developed at Pias for this very reason, to conduct research on the removal efficiency of iodine. Firstly, to simulate the highly pressurized accidental scenario, a compressor was used. The highly compressed air from the compressor was first passed through a moisture separator, then through a pressure regulator, and then through a rotameter. The air then got heated by an air heater up to a temperature of 150 degrees centigrade. The reason behind keeping this temperature is that the sublimation temperature of iodine is 85 degrees centigrade. So to keep the iodine in vapor form, the temperature of this setup is kept above 85 degrees centigrade temperature. The heated air then goes here, gets mixed with the iodine that is being pumped by a dosing pump, and the air gas mixture moves on to the scrubbing column. Now the scrubbing column contains a scrubbing solution of sodium thiosulfate and sodium hydroxide for removal of iodine, and a Venturi scrubber. Now look at the zoomed picture here. The scrubbing column, in the scrubbing column, the iodine enters here into the Venturi scrubber, and when it reaches the throat section, its pressure decreases. Now the water present in the column is at higher pressure as compared to the throat section, and this throat section contains very small holes named as orifices. So due to pressure difference, the water from the column moves into the throat region via these orifices and makes a contact with gas. When the gas-liquid contact takes place at the throat, due to inertial impaction, the liquid gets transformed into very small droplets. Now, due to concentration difference, the iodine gets trapped inside these droplets and it reacts with the chemical solution and gets retained. Now, the iodine that is left behind at the exit of the Venturi scrubber goes out into the column, gets transformed into bubbles, and due to concentration difference, goes out of the bubbles, reacts with the chemical here, and gets retained. Now, this is how the iodine removal takes place inside the scrubbing column. Now, at the exit of the scrubbing column, all the iodine that is left behind along with the air moves along this path, gets trapped inside this trap bottle that is containing potassium hydroxide solution. Now, the outlet sample is taken here and the inlet sample is taken here. This is also our trap bottle containing potassium hydroxide solution. Both these trap bottles are analyzed by using UV-vis spectroscopy to estimate the removal efficiency of iodine. This is the picture of a Venturi scrubber from our lab. This is the scrubbing column and this is how it works. Now let's move on towards the result and discussion section. Different operational parameters were varied to see their effect on the removal efficiency of iodine. Firstly, both gas and liquid flow rate were varied to see their effect on each other. When the gas flow rate was increased, it was observed that the liquid flow rate decreased. This is because as the gas flow rate increased, it made a contact with the liquid with more force and pressure, decreasing the penetration of liquid that is entering into the Venturi scrubber and hence decreasing its flow rate. But when the head of the liquid was increased, it was observed that the impact of gas on liquid decreased. As it can be seen here, at lower liquid heads, the decrease in liquid is sharp as compared to at higher heads, which means that at higher heads, liquid made a resistance against the gas with more pressure, hence decreasing its impact. So the decrease in liquid is higher at lower liquid heads as compared to as higher liquid heads. Next, the gas flow rate was varied by keeping the liquid flow rate constant. As you can see here that iodine removal efficiency was increased as the gas flow rate was increased. This is because when the gas flow rate was increased, it contacted the liquid with more force and the inertial impaction increased due to that. Due to increase in inertial impaction, more finer and smaller number of droplets of liquid were produced. The smaller and finer liquid of droplets increased the surface area of contact between liquid and gas, and due to this the mass transfer between liquid and gas increased. Now, due to increase in mass transfer, more iodine got retained and hence removal efficiency of iodine increased due to that. So, if we increase the gas flow rate, the removal efficiency of iodine will be increased. Next, the liquid flow rate was varied and gas flow rate was kept constant. As you can see here, the removal efficiency of iodine also increased with increase in liquid flow rate. This is because when the liquid flow rate was increased, more liquid entered into the throat of Venturi scrubber and more number of droplets were formed. These more of a number of droplets led to the higher mass transfer and more iodine got retained as a result of it, and hence the removal efficiency of iodine increased. So it means that if we increase the liquid flow rate, the removal efficiency of iodine will increase as well. So it is concluded that both gas flow rate and liquid flow rate play a huge role when it comes to removal efficiency of iodine. And if we can control these two parameters, we can control the removal efficiency of iodine as well. Secondly, a removal efficiency of greater than 99% was achieved by performing these experiments, which fulfills the requirement for filtered containment venting system. Now, the next step would be to perform these experiments on methyl iodide as well and to achieve the required removal efficiency for methyl iodide. Now, if this continues, this system will be part of our nuclear power plants soon, inshallah, and it will be capable of coping with any nuclear accident and for keeping the people and environment safe. At the end, I would like to thank Higher Education Commission for funding this research. This is the list of references that can be helpful regarding this topic. Thank you so much for your time. If there are any queries regarding this presentation, please let me know.
Ladies and gentlemen, she was Ms. Javeria Ahmed from Pyas, Islamabad. If you have any question, please ask your question, and you can ask it your question in the chat box.
Okay, thank you very much for joining us, Ms. Javeria. I have one question from the audience. Can you elaborate that you are using the gas, which you are talking about which gas? Is it a specific gas or it's a mixture of some gases?
No, I'm using, can you hear me?
Yes, you are audible.
I'm basically using iodine, elemental iodine, and I'm using it in solid form, and its sublimation temperature is actually 85 degrees centigrade. So I'm heating the system, and I'm keeping the system at 150 degrees centigrade temperature, so it maintains the sublimation temperature of iodine, so it transforms into vapor form, and hence I'm using the iodine in gaseous form in this experiment, and mixing it with the air that is coming from the compressor.
Okay, thank you very much, Ms. Javeria Ahad.
Thank you.
Okay, thank you very much, Ms. Javeria Ahad. Now I am moving towards my next speaker, and she is Afsana Shaheen. Her affiliation is from Bahria University, Lahore, and the title for her presentation is Optimization of Rotation of Food Waste and Fallow Dung in an Anaerobic Digestion. So I would like to invite Ms. Afsana Shaheen. Please, Ms. Afsana Shaheen, please share your thoughts.
Assalamu alaikum. I'm Afsana Shaheen from USPCAS-W, and my topic is optimization of ratio of food waste and buffalo dung in anaerobic digestion for biogas production. So this is the background. Annually, 1.3 billion tons of food is wasted, which has serious consequences on the air. So it creates air pollution. Pakistan wastes about 40% of its food, which releases greenhouse gases upon dumping openly. Degradation results in release of CO2 and methane gas, etc. Another problem is Pakistan is facing energy crisis. And this food wasted can be converted into many useful means through anaerobic digestion and converted it into biogas, which can be used as a fuel. So methodology of my study was food waste was collected from USPCAS canteen, and it included meat, vegetables, and grains. So this was then characterized by separating the bones and other undegradable things from the waste. After which a slurry was made from food waste and buffalo dung, which had different ratios. So only food waste cannot be used, as study shows that it should be used with some other core digestive material. So this food waste was mixed with buffalo dung at different ratios to see at which ratio the biogas production is maximum. So the ratios used were 1:0, 4:1, 3:2, 2:3, 1:4, and 0:1. So the substrate to water ratio here, if until this part, the substrate was there. Substrate included the buffalo dung, food waste, and inoculum. Inoculum was also matured cow buffalo dung which contained methanogenic bacteria already. So that was used in an amount of 15%, and the other substrate, further included food waste and buffalo dung in these ratios. So this was the kind of BMP reactor, and 6 of such reactors were set up parallelly, where stirrers were used to stir the substrate so that it will mix uniformly from time to time, and the stirrer used had a frequency of 60 RPM. These were the results for proximity analysis. Total solids were 16% for food waste and 18% for buffalo dung. Volatile solids were 10% and then 16% for buffalo dung. So, soluble COD was 980 milligram per liter for food waste and 1500 milligram per liter for buffalo dung. So pH was 6.4 for food waste and 7.12 for buffalo dung. And the substrate's pH was initially maintained to 7 by using 0.1 normality in AOH to neutralize the substrate. So these were the results at different ratios. As you can see that the optimum ratio here is 2:3. Food waste to buffalo dung ratio here, which is optimum and at which maximum gas is produced, is 2:3, and the minimum gas is produced at 1:0. The minimum biogas produced here is in the sample where just food waste is used. So minimum cumulative gas was produced at 1:0, which was 222 ml, and the maximum biogas produced was 414 ml, which was at the ratio of 2:3. So at 1:0, only food waste was used in the sample, and studies show that this study showed that at only food waste samples, biogas production was inhibited in between because of lowering of pH, which had to be maintained continuously or gradually. So at 2:3 pH fluctuations were rare, and the cumulative biogas produced was in highest volume, which is the optimum food waste to buffalo dung ratio. So the optimum ratio acquired was 2:3 for cumulative biogas production. So the conclusion is only food waste substrates lower the pH, which leads to inhibition of biogas production. This means that a specific pH is required for methanogenic bacteria, which can survive at 6 to 8 pH. So in this study, when we check the pH, sometimes it decreased to 3 as well, only in the food waste samples. So that's why the production of biogas in 1:0 sample was minimum. We will need to regulate the pH frequently if we want to produce biogas in optimum amounts or in increased amounts. So food waste can be used in 40% volume with buffalo dung in order to optimize the production. So we concluded that if only food waste is used, the production of biogas is not that good, it should be included with 40%, it should be included with 60% of fallow dung, and the food waste should be 40%. So that's when the biogas production is maximum. So these are the acknowledgement. I want to acknowledge US Pakistan Center for Advanced Studies in Water. This project was funded by USAID, Mehran University of Engineering and Technology, and the University of Utah, USA. So these are the references, and thank you so much.
Okay, thank you very much, Ms. Afsana Shaheen. From the audience, if you have any questions, you can ask in the chat box.
Okay, thank you very much for joining us, Ms. Afsana Shaheen. I have a question for you. Is it economically sustainable to produce this biogas from food waste and buffalo dung? Is it economically sustainable?
Sir, I had to just know the optimum ratio, and yes, it is sustainable because you know, food is a material which is which you get for free, and dung is almost free too. So the setup is not that expensive, but you can you can produce this economically.
Okay, there is one more question from the audience. Please elaborate on the pH related to sample 2:3 ratio. It seems too much dung, 60%, and substrate to 40%.
Yeah, it is, too much dung because, when food waste is used, so pH is reduced when a lot of food waste is there. So this was the ratio I got from the experiments I performed. So my basic purpose was to find the ratio, and the ratio was 2:3. So that's how it works.
Okay, thank you very much, Ms. Afsana Shaheen, for joining us.
Thank you so.
Okay, thank you very much, Ms. Afsana Shaheen, for your presentation. Now, our next speaker name is Dr. Amir Rashid, and he is from ComSat University. The title for his presentation is Unmanned Autonomous Ababil System for Seed Plantation Utilizing Spectral Camera Analyzing Using Drone Technology. So I will request Dr. Amir to please share his thoughts.
Assalamu alaikum. My name is Dr. Amir, and I'm an assistant professor in ComSat's Electrical Engineering Department, Islamabad campus. First of all, I feel really proud and honored to be a part of this international conference on energy, water, and environment that will be held on 31st of March. And once again, thanks for considering our little work. It's a great moment for me in a way. The UET is basically my alma mater. I have done my graduation and my master's from this institute. For it's a very thrilling aspect in my life, and it played a pivotal role, particularly in developing my little knowledge. So basically, our paper, based on it's an unmanned autonomous Ababil system for seed plantation utilizing spectral camera analysis and by using the drone technology. I, along with my colleague, we have worked a lot on this and have gained so many national, international award, particularly working on this particular subject. We have also won funding from Ministry of Information of Technology. It's in Pakistan department. And so far, one of the other aspects is this: we are also, you can say, we have crossed one level of Turkish competition that will be held in particularly in coming weeks. So my team is very determined, and we have done a lot of work on it. So let's, if we talk about the trees, or if we talk about our environment, with the passage of the time, the whole of the world, or you can say the people, they have progressed from the cave life to our modern life. But the main thing is this: the environment, they have paid a very big price, particularly in maintaining our life, or you can say in developing our life. We developed our houses for our living, and what we have done, we have cut so many trees, and that's you know, we get the trees we used in our life for burning and everything, and then you know, we have developed our life. For that matter, the deforestation process is, it's very at a high level. Initially, there were so many jungles are available, so many woods available, and the environment is available. But because of this deforestation, you know, we have lost our environment. The global warming is comparatively is very high at the moment, and we can see that the glaciers and the other impacts on our life. And you know, there is also a very big part, we are losing the nature, the loss of the biodiversity. So we have very bad impact on our earth because of the cutting and because of the deforestation. So that the thing comes in our mind, that how to solve this issue. For that matter, we have thought of Ababil system. Ababil, basically, it's as being a Muslim, we know that, you know, it's a small bird and have a very you know, it's a history. The Ababil means it's becomes the shooting something from the sky and all. So for that matter, we basically work for this purpose, and for even the food insecurities. So what we have done, we have used a drone technology for the seed, you can say seed disperser. For that matter, we have you know, we use the spectral camera of the drone, and then we acquire the image. After acquiring the image, we process the image sorry, where we have to plant, you can say where we have to sow the seed. Basically, it's a total environment. Within the total environment, even where the trees are there, and if we want to add a seed, we don't want to disperse a seed on a tree. We want to find out where the space is, you know, where the trees are not present. We find out, and then by the image segmentation, and then by the image extraction, we just throw the seed, or we shoot the seed at that particular moment. For that matter, we have a 3D map of an area of, you can say, where we have to plan a mission. Before that, we have a plan, and you know, the drone will roam around and will get all the data where we have to put our you know, seed. And even we can find it out the soil fertility, that whether the seed will be penetrated and or not. So this is you can say we have worked on it, and as you can see that, you know, we have a map, and we have a 3D mapping before even launching the drone or for the dispersal of the seed. Now there is a little specification of our drone is there. Without, you can say it's a payload. Payload means, you know, the seed weight, or that's a 950 grams, and total payload that our drone at the moment that can carry, it's around the 3 kg, and then your maximum flight would be 30 meters, that is without payload, and with the payload it's a 20 to 25, and then we have you can say a speed of a 45 kilometer per hour if we don't deploy the payload, because for the drone will go first, and then, you know, roam around the area and capture the images. And if we have a payload, then it will move on around a 30 kilometer per hour or so. And at the moment, we have planned for a 1 kg, which I'm extending at the moment for the next phase. And that is a 1 kg payload at the moment. And with that is a 35 kilometer. These are some aspects or aspects of the, you can say, our drone. Then again, by driving, or you can say by before going out our drone, we have to find out whether the outward weather is good, or what's the temperature outside, what's the wind, whether there is a windy or you can say there is a gust or whatsoever. So all the data before launching the drone, we have our own mobile, but the developed mobile application. So we knew everything about, or the accuracy level can be improved, whether if we will send our, or if we will shoot our drone in a windy environment. So there is strong possibility that when it will shoot the seed, it will not go on a appropriate or a proper field. So we gather all the data, the visibility and other aspects. We have application that can show about all these, you can say all these features and the characters we know already about the environment we are sending our drone. And if you talk about the results, we have also you can say tree calculator, or where we can add our what we have our, like the, if the drone is moved like 25 feet or 30 feet or even the 10 feet, depends upon, you know, the terrain where we are sending the drone. And then we also put whether we have to put the seed in a 1 feet or a 2 feet environment, or probably 3 feet. We can automatically you know, calculate. For that we have a calculator, and then we put all the values, and then we can find it out whether the approximately how many you can say seed we have to disperse in which environment. So for that you can see that, you know, it's around the number of you get the seed we can disperse along with the area, and then we find out, we can plot a graph that how many seeds we can disperse in that particular time and the area covered in that way. So what I'm trying to, it's basically the main conclusion that we are really trying to find it out. Basically, it's the biodegradable seeds bullets, and that we shoot in it, because if we only send the seed, probably it will drop at a place where he will never find out the water and the other thing. So we are trying to encapsulate the seed in a biodegradable you can say bullet, and then we shoot it. So the loss, you can say the damage of the seed we have calculated so far is a 5 to 10%, but you know, the more appropriate is a 5%, 7%. We have tried and we have find out you know, our data that it's around 5%. And then what the system that accurately detect also the fertility or the nutrition of the composed soil. How we have, you know, the images, and with the help of the images we can find it out whether the soil is more fertile or not. Yes, at higher places where the human approach is not even possible, but we can somehow we can calculate that whether the terrain is good enough for the seed that can penetrate or it will not penetrate. So basically with the help of this technology we can also find it out the terrain state has. And then the next thing, that is the more major part, that it can reduce the manual work, because if we count that a person or any labor can you know, the farmer can go or the plant of the seed, it's very less. And where the terrain is very difficult or we can't approach there, it's very almost impossible to reach there, and then we can plant it. So for that matter, the drone can go automatically, and then our, you know, our designed or our desired place, find it out the places, and then, you know, the shoot one. So it is a very good you can say that is the main reason that we can deploy seed or disperse seed in any weather or even in a poor infrastructure of the roads or anything. Again, the other thing is it can you can say it's a very eco-friendly because it is not because of that, you know, the dispersal of the smoke or anything. We can charge it and then we can send it at our proposed you can say monitoring path, rather than, you know, we have also an option that if the drone will go away our range, that it can automatically come back. So basically we prevent, in fact, also the illegal cutting, because if you keep on watching, and then we can find it out that the once the trees are there, and the next day or the after week we have another survey that it will tell us by comparing the previous videos that, you know, the illegal cutting is there, then we can take appropriate action, or we can inform the law enforcing, or maybe the forest ministry, that, you know, the things are happening. that things are not happening good. And even if the wild animals are, they are going to be less, or you can say because of the habitat change, then even we can also find it out. And the other thing, we can also provide help to the tourist, or you can say the mountain climber, or that can give a very good view to the tourist to come over this place. I will thankful for the acknowledgement of the J&J company and the FAMES, one company FAME, one is you can say one of my own company, you can say, and they are basically even helping us or providing us funds and all that stuff for the research. I have, there are so many references, but for this one, 5 or 6 references or the books, I have put it over here. Thank you, and once again, thanks a lot for giving me this opportunity to share our work over here. And I'm really hopeful for further, whatever the feedback I will receive, I will try to improve my present research. Thank you. Assalamu alaikum.
Okay, thank you very much, Dr. Amir, for your presentation. From the audience, if you have any question, you can ask in the chat box.
Okay, thank you very much, Dr. Amir, for joining us. It's a wonderful presentation and wonderful idea. As automation is the step towards the Industry 4, and which is the ultimate goal for this era. So my question from the audience is that, is this system currently used anywhere in the world?
Assalamu alaikum. First of all, thanks for, again, I'm very thankful for giving me this opportunity to talk my alma maters. Yes, of course, there are so many systems, particularly if you talk about the drones, they have been underutilization, particularly in America and Switzerland and other places as well. But the main thing is the development of the gun that we have used for the firing purposes. Because all the best thing is this: the drone that we are using, you can say I have managed to make within 2 to 3 lakh. But the same purpose, the same size drone is available in the market that is more than 2 million RS. You can say it's around you can say 2.5 to 3 million, if I'm correct. Because I managed to secure the funding of Always PSA, Pakistan Science Foundation, and the next step is move toward the spray. The dispersal purpose is the spray on the crops. Whatever the other companies are doing in the world, they are providing the same services, or not the same, the less services even, with a very high expensive rate. So the main thing, or the main objective of jumping into this domain, because in our country there is illegal cutting, and there are a lot of happening in our south. So even the KPK government, or the you can say even the Kashmir government, they have contacted me to work on that. So what I'm trying to say over here, it's not, you can say, the new idea. The idea has been already under consideration in so many countries, but by using our own understanding, or our own Pakistan area, we have modified it, and we have developed the new the problems that we are answering, the new problems that happening over here. Even the tourist spots we are covering, and then we are covering about even the animal habitat. Also we are covering that, the people, even the lost people, particularly in the river and all that. So the spectrum, or you can say this project is a very broadened, you can say the limitations. What I'm asking even to my all audience, that please come and join this particularly domain, because many government funding are available for this one, and they really need our, you can say, our contribution towards this one. If we are managed to spread seeds, like in a day, if we can manage to spread seed for 4,000 to 5,000 per day, which is almost impossible if you talk about the human capacity, or the farmers, or the other one, because 80% of our country is based on agriculture, or we are somehow related with the agriculture sector. So this sector is, unfortunately, is very far away. So my aim is to give the prosperity to this country and give, you can say, a lot of further ideas to our audience and even my university. So we have to work on it. Thank you. Hope the question is answered.
Thank you, sir, for being us.
Okay, thank you very much, Dr. Amir. Now, the name of our next speaker is Miss Sadia Hashmi, and she's from Bahria University, Karachi. The title for her presentation is Industrial Dye Decolorization by Partially Purified Microbial Legacy from Bacillus sp. So I request Ms. Sadia Hashmi to please share your thoughts.
Bismillahirrahmanirrahim. Assalamu alaikum everyone. I'm Sadia Hashmi from the Bahria University Karachi campus. I'm going to present today in ICEWE conference. My research topic is about industrial dye decolorization by partially purified microbial lacase from Bacillus species. Because of the rapidity of the industrialization and the urbanization, it results in the large amount of the waste into the environment, which in turn creates more pollution. Environmental cleanliness and the sustainability is crucial for the human well-being, but modern-day industrial pollutants, as industrial pollutant that affects the air, water, and the soil, it can directly as well as indirectly compromises the human health and the environment. So there are the different applications of the dye are used in the different industrial sectors, like in paper and pulp industry, in the textile industry, and also in the food industry. There are the many methods are available to treat the dye effluent, like physical and the chemical methods are available, but they are not the economically feasible and the environmental friendly method, because at the end a large amount of the sludge is produced, which in turn itself requires a treatment. So here I use a very eco-friendly source, that is the microbial source, in order to decolorize the dye by doing the certain methods. Like for my first step is to isolate the dye degrading bacteria, and in order to and then for going for the production of the lacase, screening of the lacase production. Then after the isolation, the certain microbial and the biochemical characteristics were carried out in order to identify the species of the bacteria. Then after that, the optimization of the certain physical and the chemical parameters were carried out, and the last one is the partial purification of the lacase was done by doing the gradient soil precipitation technique. Now I would like to discuss the result of my research study. The soil, it was collected from a very famous textile industry soil. Here it is located in the Karachi. A few grams of the soil sample was taken and then inoculated in the flowers which contain in the broth medium, and then placed in the rotatory shaker for about 72 hours at 50 rpm at 30 degrees centigrade. After that, the culture medium was treated onto a neutral agar plate along with a certain dye concentration, and the colony which showed the maximum degradation zone was carried out for the further study. Then after that, the certain biochemical characteristics were carried out in order to identify the bacterial species, which is the Bacillus subtilis. The next step is after the isolation and identification and characterization. The next one is the select the fermentation medium, which is very much important medium. So the 5 different reported medium were chosen for this purpose, and out of the 5, I found the medium 3, which is actually the personal and house medium, is considered it showed the maximum production of the lacase to decolorize the dye, which is about the 90%. Was noted. So this medium 3 is considered as an the fermentation medium, and on this fermentation medium there were certain parameters were optimized, which include the physical and the chemical parameters. The physical parameters include the time period, the temperature, and the period, and the chemical parameters will includes the carbon sources, the nitrogen sources, and the mineral salts. So first we will discuss about the optimum time period. The incubation time plays a very much important role in secretion of the enzyme. For this, the enzyme activity was performed by using the glycol as a substrate, and the decoration rate was found out. So the result it showed that malachite green it showed the the optimum time for decolorization. The optimum time for the production of the lactase to decolorize the dye was found to be after the 24 hours of the incubation time. Then is the optimum temperature. Temperature has important influence on decolorization of the dye. After performing the enzyme activity, I found that the bacteria showed more production of the lactase for decolorization of the malachite green at about 50 degrees centigrade, while the further increase in temperature, it does not show any increase in the decolorization rate. Then is the pH. The pH, as you know that here I want to add it that the bacterial soil which I'm using is the bacteria is the Bacillus subtilis, and from that bacteria I used to extract the enzyme which is the lactate. So we all know that enzyme they are the sensitive for the temperature, the pH, and also the time. So to provide the maximum production of the lactase production, these usually optimization parameters are carried out. So different pH ranges were selected, and the pH 5 is found to be the optimum concentration. To on this optimum concentration, I found that lactase production was shown the maximum enzyme activity to decolorize the dye about 80%. After all the physical parameters were optimized, the next is the optimization of the chemical parameters, which include the carbon and nitrogen sources and the mineral salts. Here the carbon source is the glucose source with the different concentration of the glucose concentration are carried out, and that which is considered as the 0.5 concentration is considered as the optimum concentration of the carbon source in the fermentation medium. It showed the maximum decolorization rate about the 99%. Then is the nitrogen sources. Here the two types of nitrogen source are used in the fermentation medium. One is the organic nitrogen source and an inorganic nitrogen source, the yeast strain and the ammonium nitrate. So both of the optimum concentration for both of the parameters are the 0.5 considered, which show the 80 and the 61% respectively. Then is the optimization of the mineral salts. The calcium chloride, magnesium sulfate, and dipotassium hydrogen phosphate were used within different concentrations, and as we can clearly see that the 0.1 concentration of the calcium chloride and the magnesium sulfate, it showed the maximum decolorization rate. The further increase in concentration of the salt in the fermentation medium, it does not show the high decolorization rate used to decline. After all the optimization of the physical and the chemical parameters, the next one is the optimization of the dye concentration, which is very much important. It found that 40 microliters considered different dye solutions were prepared, and found that the 40 microliters is considered as the optimum concentration in the fermentation medium, while further increase in dye it's used to decline because of the resistivity nature of the dye. This is the final concentration of the optimized medium component, which is actually the newly invented reported medium. I'm going to report this medium in my research article, hopefully. Now I would like to conclude my topic. The dye-degrading bacteria isolate from the industrial soil and is screened for the lactase. The bacillus satellite, it produced the lactase in maximum concentration with Bushnell and Haas medium at 40 degrees centigrade, and the pH is about 5.0 after the 24 hours of the fermentation period. Glucose, yeast extract, ammonium nitrate, dipotassium hydrogen phosphate, calcium chloride, and the magnesium sulfate has promising effect on the lactase production. The 40 microliter concentration of the dye, it showed the maximum liquidization rate, which suggests that lactate, which is produced by the bacillus satellite, has an ability to hydrolyze the malachite green effectively. Now, at the end, I used to thank my University of the Karachi, which is my parent university, from where we have done the research article for the Ample Research Award, and to my supervisor, Dr. Sharul Kader. He's my mentor and my supervisor. I really was thankful for him to providing me and giving me such a great knowledge about the bacterial source to degrading the bacteria, to degrading the dye. And I'm very much thankful to the Bahia University to giving a platform in order to present myself and to affiliated with the university to present myself in the ICEWE. These are the certain references which I used in my in this presentation and the abstract.
Okay, thank you very much, Ms. Sadia Hashmi. From the audience, if you have any question, you can please ask it in chat box. Okay, thank you very much, Ms. Sadia Hashmi, for your presentation. On the behalf of Organizing Committee of ICEWE 2021, I am thankful to the session chair, Dr. Tayyaba Asif, all the presenters, and the participants. It was a wonderful session, and I hope the audience had enjoyed it. We have other sessions running in parallel and the exhibition as well. So enjoy our parallel sessions. Thank you for sparing time for this productive session.